Okay, well, welcome back, everyone. Today, we're going to switch gears a little bit and start talking about within-host viral dynamics. So until now, we've been talking about epidemiology with the exception of a few minutes in the first lecture. This time, we're now going to be focusing not on what happens in a population of people, but what happens inside the body of an individual. And that's typically called within-host viral dynamics models. And there are a couple of housekeeping things I wanted to get out of the way. Having had a conversation the other day, I strongly recommend that if you haven't read it before, that you take a look. I loaded it into the student materials folder under the papers. This is an old article from 1989. It's actually even older than 1989 because it was actually recorded in the 50s. And only published in the 89, called Pathological Science. It was published in a popular science journal called Physics Today. It was written by Irving Langmuir, who's a Nobel laureate, about how to know whether a scientific article is reasonable or not. And I gave you some readings from Yi about how to sort of evaluate a mathematical biology article. In the history of science, there have been many non-existent discoveries. Sometimes those non-existent discoveries are fraudulent. At other times, they are the result of wishful thinking. Sometimes they're legitimate mistakes. But one of the things that's quite interesting about them is that quite often there will be an announcement of a major scientific discovery. That scientific discovery will be replicated by large numbers of people. And then after a while, there'll be some doubts about the legitimacy of that scientific discovery. And eventually, there may be some refutations and maybe even a retraction of the original work. But in any case, it winds up that whatever was discovered didn't exist. And that happens in all fields of science, but biology seems particularly prone to these, perhaps because making advances in medicine is financially remunerative, and therefore there's a strong element where people want things to be true. So if you look at the popular literature, even the scientific literature, and ask how many great advances in the treatment of cancer have been published, we could improve the treatment success for cancer X by 90%. And then you look 10 years later, are we using that treatment? The answer usually is no, we're not. Well, something didn't add up. Sometimes, as I say, there are many reasons for that. Langmuir was focusing on physics, non-existent discoveries, of which there have been many. There were the mythical N rays of the early 20th century. There was polywater. And this article was published in 1989 because of the whole cold fusion fiasco. I don't know if any of you ever ran into cold fusion, but there was a claim by a group at the University of Utah, ironically when I was visiting at the University of Utah, that they had discovered a way to create fusion in a test tube. And as you know, if we had nuclear fusion, which is what powers the sun, there would be no energy problem. And so if you could do nuclear fusion in a test tube, the world would be totally different because you'd have an unlimited source of energy for free. And so there were a lot of people who really wanted that to be true. It wound up not being true. And this article from the 50s was found and resurrected in a response to the cold fusion hype. But there are many different examples. And in some courses over the years, I've had modules where we studied this problem, and there are books also on it. Some of these books are focused on physics, some on chemistry, some on biology, some on engineering. As I mentioned, there are plenty of various kinds. But Langmuir has some nice things to think about when you're reading an article. Does it claim is it showing something that's barely detectable? In other words, are you seeing something in limited resolution? Most new scientific discoveries are of that kind, or many are. The reason that they're being discovered is that they're hard to find. So just because it's something that's a small effect doesn't mean it's not real. In the case of cold fusion, the effect was very large. Another great physics non-discovery was the magnetic monopole, which is also a big signal, but that's one possibility. And then the question of repeatability. One of the big problems in biology is that experiments are not repeatable. Some lab does an experiment, they claim to have a big signal, other people do the same thing, and they don't see it. And maybe it's because it's not there, or maybe it's because there was something they were doing specifically. Their particular strain of animal, their particular reagents were special. And then if things go against common belief, if it counters the textbooks, it doesn't mean it's wrong, but you have a slightly higher standard of proof if that's the case. Criticisms met by ad hoc excuses. That certainly happened with cold fusion. When they started to fall apart, they came up with all sorts of ways of justifying it, including suing the people who published papers refuting it, by the way. The president of the University of Utah in the end had to resign because he was using the legal department of the University of Utah to sue critics. Maybe it sounds familiar. And then that some people like it, some people don't, but it never is completely accepted. And so in our discussions of epidemiology, I think we're running into some of these issues. And so I thought it would be worth drawing your attention to this. It's a short article. It's a good read. And I'm sure when you read it, it helps you be a little bit more critical and a little bit more skeptical when you read things. And I promise you, in the area of biology and bioengineering, there are lots of these. There are plenty of discoveries that we're going to cure cancer X. There are plenty of discoveries that we've invented ways to induce stem cell regeneration, tissue engineering. These are all areas that are full of these kinds of things. The flip side is that sometimes the discoveries really are there. A lot of our understanding of cancer biology in the textbooks was wrong. RNA interference was a key mechanism of regulation in cells that was missed until relatively recently. And so just because something is contrary to dogma doesn't mean it's wrong, but it does mean that you should be skeptical. And so I really do encourage you to read this article. We were talking about it with an undergrad in the department, and I think you'll enjoy it. So I wanted quickly to get updates. I'd ask you to resubmit your project proposals today. I hope people were able to do that. And I wanted to get just a quick update. You're welcome to slide share if you want. You don't have to. If you want to share slides, let me know and I'll unshare. Reshare your screen. Just to get an update on how you're doing in terms of defining and getting started on your projects. And so I'd like to quickly go around the room and hear about that from people. Again, if you want to tell me, if you want to screen share, that's fine. Just let me know. If not, that's fine. Why don't we start, Tyler?
Yep, can you hear me?
Yep.
Okay, so I've maintained the same idea. I'm going to do the in-host viral kinetics with the immune response as well. Mm-hmm. And I've done a little bit more research on it, and I want to specifically do either hepatitis C or H1N1. And I want to basically make the model of the immune system, the general response, and then the general or dynamics of either virus, and combine the two models and see how that works. But that's pretty much as far as I've gone as far as updating.
Okay, great. Well, we're going to start doing some of that today, so I hope that helps refine your ideas and go forward with that. That's fine. Antonio?
Yeah, good afternoon. So I have decided to switch my focus to exploring the effects of like preemptive antibiotic treatment for bacterial infections. So I found a few good papers that model the effects of like standard antibiotic treatment on bacterial infections, both in populations and in individuals. And so I kind of want to start with replicating those models and then adjust the model to perhaps if the population or the individual had an initial dose of antibiotic before the infection even started, and kind of compare how antibiotic resistance builds between the two of them, and how the disease spreads among a population or an individual.
Those are great topics. Antibiotic resistance is a huge problem. Pre-treatment at low dose is a huge problem, not only in humans, but also in animals. One of the things that you have are growth enhancers, which are low levels of antibiotics. For some reason, you give cows and chickens low levels of antibiotics, they gain weight much faster. They also develop antibiotic-resistant bacteria much faster. And so it's not clear why the antibiotics help them. They're not at levels which would prevent disease. And so that's an example. And so I think that's great. I think if you can try to, in the next, as soon as possible, try to write up a little summary of that so that we can begin to focus that, because that's a really important but also very large topic. And so the key from my point of view is making sure that you have something that we can make sure it's not too, at least the beginning part, that's not too ambitious, but that's a really interesting topic.
Will do. Thank you.
Devin?
Yes. I thought we were supposed to make slides, so I'll go ahead and share those if that's possible.
Sure.
Okay. So I'm planning on sticking with the same topic, the oncolytic viruses that kill cancer. I started to look more into the math that the reference paper used. And so it's kind of like a predator-prey relationship that this one uses, where the change in uninfected cells is represented by the number of initial uninfected cells times some sort of cancer growth and death process function minus the virally infected cells here. And then the change in infected cells is represented by the rate of the viral infection minus the viral infected cells that end up dying from the population. And so currently, the exact form of the F and G functions aren't well defined, since they haven't been extensively researched, or they might just vary depending on what type of cell or virus you're looking at. So I plan on looking at different functions to determine like what sort of virus would best eradicate the cancer cells. So some of the cancer growth functions, there's exponential growth functions where F is just 1, there's logistic growth functions, and then it's got some biological constraints that need to be followed. And then it's kind of the same here for the virus spread function G. There's some biological constraints and some different options that could be used. And thinking about extensions that I want to use for my project, I want to potentially add a slider in Tellurium for some of the variables that can be adjusted, like starting cell population, stuff like that, viral spread rate. And then potentially make other cancer growth functions and virus growth functions according to other research. And I've stated some stuff here. And that's all.
Oops, I think we lost you there.
That was all.
Okay, that's great. That's very nice. It might be worth talking to Mercedes a little bit, because she's also interested in cancer modeling. And so I've been talking to her over the weekend about possibly finding some ODE models of cancer and immune response. And so your problems aren't identical, but my guess is that if you communicate a little bit, you might be able to exchange some useful ideas.
Okay, thank you.
Brian?
Okay. So basically, I've kind of kept with that main idea of essentially seeing how like a SARS-CoV-19 vaccination would help out in terms of like modeling the like a vaccination and like the efficacy of it. I kind of took some of my old ideas and I combined them together. So after I believe the last couple of lectures, I saw that there was a pretty easy way of implementing herd immunity into it. So I implemented. I'm also planning on implementing like herd immunity, seeing like what vaccination rate that we would need in order to have herd immunity. And I've done a little bit of research in terms of finding a couple of papers that I think would be useful as sources. And yeah, the only other thing that I would mention is that I was thinking about switching a little bit last week because I was reading the one paper that we had to do for class, which was over the RSV virus, and that specifically targeted, I guess, the age-related disease in terms of how it related to children that were under 1 year old versus the rest of the population in Valencia. And so I thought that was kind of interesting. Age relation in COVID-19 would be a kind of interesting thing to model, but if that doesn't fall through, then I'm just going to continue on with this track with vaccination.
Okay, maybe we should jump because Kali, I think you had some ideas that were somewhat similar to that.
Yeah, I'm also looking at a COVID vaccine model.
So you're welcome to work independently. You're welcome to pair up if you prefer, but I'd suggest maybe you can share some ideas, share what you've been reading and what you've been thinking about, at least possibly. Kali, did you have any more you wanted to say, or just update us then on where you are?
Yeah, I'll share my screen real quick.
Okay.
So I found a paper recently published that talked about an SVIRS epidemic model for not a specific disease, just it's a math and engineering paper, so it's a bit more generic. I thought it would be interesting to look at. And even though it doesn't have like death included in the acronym here, it actually does include death in the model through these terms here with the death rate from natural causes and the disease. So it actually is like an SVIRS-D model, which we discussed yesterday might be like a good place to start with this. So this model is a little bit more complicated. As you can see, there are more equations here. It also introduces this idea of a latent period, right, in the model. So it's like, I guess, sort of an SV. It has a little E term in there in disguise. And because of that, it uses these terms like S of time t minus the latent period tau, or I of t minus the latent period tau. And I think that's just like a difference in the number of susceptible, infected, or vaccinated people at the current time versus like the beginning of the previous latent period or the latent period before this time. And I'm still trying to figure out how/if I can do this in antimony. But here's what I have so far. I don't know if I should just delete this latent period consideration for now.
So delay equations, antimony intrinsically doesn't do delay equations.
Right.
And the reason is mathematically delay equations are hard to solve. And there are relatively few. MATLAB has some general purpose delay equation solvers, but in general, you need special methods to solve. There aren't generic solvers that work with all delay equations, and so to have explicit delays winds up being difficult. There are techniques to approximate delays, which is you introduce a box between the thing that's starting and the thing that's delayed, and then you can interpolate delays. And I can show you probably offline how to implement delays if you need them. One thing to do is to start with those delays set to 0. and see how different the results are. And then if you want to implement delays, we can do it. I can show you how to approximate the delays. It means you won't get the exact results that they got, but you can get arbitrarily close to the results they get. Essentially, what you do is you build a bucket brigade where you pass whatever the thing that's being delayed is from one box to another box to another box to another box.
Mhm.
And as you add more and more boxes, you get closer to the exact delay equation. It doesn't look terribly pretty in your code, but it works fine. And actually, some of the things that I'll show you in either today or next time have the form they do because they started out life as delay equations that had to get undelayed.
Okay.
So that sounds quite interesting. And actually, we've worked with, I guess, Giuliano and Josh have worked with somebody in the med school in the Department of Pharmacology, Tarun, and he started out with a bunch of delay equations, which we had to unwind. So Josh and Giuliano have some expertise in how to deal with that. Again, it's a little cumbersome, but it's not intrinsically difficult. And very often you find that those delays are not that significant. When they are, and then you have to think about it. But starting with the equivalent thing without that delay probably is not a bad way to go.
Okay.
So that's great. And the fact you've got it coded up already is impressive.
Yeah, I got it coded up as much as I could. I still need to look through to see what these exact parameter values are.
Right.
But
Great.
yep, that's
Okay, neat. So again, maybe if you're willing to talk, there's no obligation, but since we have two people thinking about cancer and two people thinking about vaccines, I encourage you to talk to each other. I'm not saying the talking could be that you decide that you don't want to talk anymore, but I encourage you to at least talk once and think about what you found and how you're thinking about the problem. Great. Okay, Jeffrey, I guess you're the last. I don't know, Mercedes isn't on the call today. So Jeffrey, how are things going for you?
I made some slides that I can share also.
Great. You should be able to do a slide share, I think.
Okay, so this is just like a kind of description of, I guess, the basic units of these of the model. So it kind of breaks up different regions of the brain into these columns. It's based on, I guess, certain inputs affect certain groups of neurons, and they're all kind of grouped together so that it's not a really clear cut or like hardwired structure. It's more so just to make modeling simpler. And then these are the basic equations of the model, where these VIs are the potentials of the different columns. It's basically just I can't remember, it's the main kind of neural equation, Hodgkin-Huxley.
Hodgkin-Huxley, yeah.
Yeah. And then they also model some of the inhibitory neurons. In this, I'll come back to that. So what some of the previous papers have done is the model I just showed is based on a study for modeling seizures and like neuron synchronization during seizures in epilepsy. And what in the paper I got the model from, they focus more on theoretical work with the dynamics. So I was thinking what they did with these equations is they kind of changed these deltas to change the firing rates. This is, yeah, I wrote that. But they changed the firing rates to see how that affected the dynamics and kind of like the attractors and limit cycles of the system. So what I was thinking is maybe trying to apply this model to there are medications for epilepsy and that act and kind of block certain ion channels. So I was thinking changing the this M variables for different channels to kind of simulate medicine blocking the channels. Might be something to do. I'm still not so sure where. I mean, this is probably a decent idea, but some of the other ideas I've said are maybe too complicated. So I just I kind of went with. I'm not so sure how to like modify and expand upon something. So this is just my idea right now. Just something to do. I think that was it. Yeah, here are the papers I wrote. Got this from. But
So one thing you could do is start with just one or two oscillators and couple just two oscillators together and see how they behave.
mhm.
And then maybe add a third one so that you could build it up slowly. You build one oscillator model, sort of a single neuron model, and you couple a pair and do that. The issue of epilepsy is always interesting because, I mean, at some level, what a thought is in your brain is local synchronization of neural activity. If you start at the start of a thought, you have diffuse, unsynchronized neural activity, and then you get some region of association cortex that has local synchronization. And so that entrainment and synchronization is more or less what we think of as a thought, at least according to a lot of the people who work in the area. On the other hand, if you have very large scale synchronization and the synchronization is too strong, then you get the kinds of massive oscillations that you see in epilepsy. And so the big regions of the brain do have this sort of funny balance where you have to be able to entrain and synchronize in order to be able to think. But if that entrainment and synchronization gets too strong, your brain stops working. One of the paradoxes of epilepsy is what's called kindling, which is that when you have a seizure and you get that kind of entrainment, it tends to reorganize the network so that it becomes more likely in the future. So each time you have a seizure, the probability of a seizure in the future and the magnitude of the seizure when you have it are worse. That may be a little bit outside of the particular thing you're looking at here, but it's something that's a sort of a characteristic of epilepsy. Now, people like Eshel Ben Yaakov and Kunihiko Kaneko, among others, worked on that kind of synchronization problem and its relation to epilepsy. I know there's been a lot of work on that topic recently. He's at Scripps. Trying to think of, there's a very famous theoretical neuroscientist at Scripps whose name is escaping me at the moment. There were two teams. There's one at Scripps, and the name is not coming up. Failure of synchronization in my brain. And then there was also a group at Yale that was working on that. So those are great problems. But again, I'd encourage you, since we're getting near halfway through the semester, I think it's important to define as soon as possible, define a simple problem, get it coded up and get it running so that you have some time series to look at. And then you can definitely work on refining it more. I think it's a very reasonable problem. Just doing Hodgkin-Huxley and seeing how it behaves is already rather interesting. And doing Hodgkin-Huxley in a couple of cells and coupling it is also quite interesting.
Okay.
So I think that's fine. Okay, great. Well, thank you very much, everyone, for the updates. Please, please keep pushing ahead as much as you can. And on the next week or two, I think people should really try to have something coded up. Because otherwise, again, my worry is not that you're not able to do this, but rather that it's very easy to get into a situation where time runs out on you. And over the years, there have been a lot of people who took incompletes to finish up their projects, and I suppose that's always possible, but for your sake, it's better to get them done so that you have something to do next semester that's not your work on the previous semester. I had a roommate many years ago when I was a graduate student who had his degrees in order were PhD, BA, BA, PhD. And the reason that he got his PhD before his bachelor's degree was he had so many incompletes for his undergraduate degree that it took him actually several years after he finished his PhD to finish all the incompletes. And so it's the only person I know who has a PhD in mathematics before a bachelor's in mathematics. So I encourage you to avoid that precedent, although he's a professor at McGill now, so didn't hold him back. So you can take whatever lesson you like from that. Okay, great. So let me come back. So today our topics are going to be within host viral replication models. We'll start out by thinking about viral production and a little bit about cell death and then immune response. And to begin with, we're actually going to do the same thing we did in the past couple of classes, but just look at some data and think about how to analyze it. And so before that, let's just remind ourselves what we did last time. We talked a little bit about SIR models. We did simple stochastic models. I would have liked to spend more time on that in class. You're going to get one homework assignment, which will be to explore a little bit more about stochasticity in that simple model. I hope it's not too painful. But there is a fundamental question, which is when you go to these stochastic models, if you average over large numbers of replica, do you recover the ODE model or not? And in some cases you do, and in some cases you don't. Sometimes the stochastic models are fundamentally different in some way from the ODE models. And in that case, if you use the ODE models, you may misunderstand how a system works. Gene regulation in the context of differentiation is a classic example of that. The stochasticity of gene regulation means that the classic models of differentiation, which we use all the time in models of development, don't actually work. So it can be serious. We also introduced the concept of a fixed point. Fixed point is a situation in which you have some set of variables at values where they don't change in time. So if we have everything written as ds by dt equals something, di by dt equals something, if d by dt of everything is 0, then you sit there. That's what a fixed point means. That's what it's called fixed. And so ultimately, that means that all of your equations have to be 0 on the right-hand side simultaneously. So for example, for our SIS model, ds by dt was beta I times s over s naught plus mu I, di by dt beta I over s naught times s minus mu I. We set the left-hand sides, we set the right-hand sides to 0, which is our fixed point, and we can solve that one directly. Use a star to indicate the fixed point. S star is mu over beta times S naught. That's the fraction of the population that stays uninfected at any given time. I would be 1 minus that times S naught. And so in particular, if mu over beta is bigger than 1, that is R naught is less than 0, then the number of infected individuals goes to 0. That's a simple example. It also means that as I change R naught from being less than 1 to greater than 1, I go from having one fixed point, which is nobody infected, to a fixed point, which is the endemic, stable fixed point, which is the endemic. And that change of stability of fixed points is called a bifurcation. And we're not so far talking in detail about the mathematics of bifurcations, but we will talk a little bit about it. I also introduced some syntax, which we wound up not using because we didn't do the paper that needed it. Just reminding you that when you're writing antimony, if you want to do something at fixed times, there's something called an event, which is like an if statement in a procedural language. You can name them at time greater than 15, a equals a plus 15 works. The argument of the at, like an if statement, doesn't have to be temporal. It could be at susceptible population less than 26, do something. The thing that you have to remember with antimony when you have events is that if the argument of the event goes from false to true, you do whatever is on the right-hand side of the event. But you don't do it more than once if the thing stays true, and you don't do anything when the argument goes from true to false. And so if in the case of time greater than 15, that only goes from false to true once. But if I said, if sign of time positive, then every time I go through one cycle, so the time goes from positive to negative and back to positive again. Every time it goes from false to true, it'll trigger again. And so I can also have periodic triggers or triggers that occur under certain conditions. You can build fairly complicated rules that way. The other thing is that if you want to write functional relationships that change in time, if you use K2 equals 3 times K1, that's evaluated only at the beginning. And so K2 will have the value of whatever K1 is 3 times the initial value of K1. If I write K2 colon equals 3 times K1, then if K1 is changed, K2 will also change. And so that's how you define functional relationships. And those things may or may not be things we need as we go forward. We'll certainly need them at some point as we go forward, but I don't know exactly which class we'll need them in. But I wanted you to just be aware of those. Again, antimony and tellurium have lots of features which are documented in the manuals online. We'll talk about the ones that we run into, but you may need, especially for your projects, you may find that there are others that are useful. Okay, so now I want to come back to something we actually started with in the very first class and then moved away from to do epidemiology, which was building a very simple model of viral infection. And I'll very quickly review the slides that we went through in the very first lecture because it's long enough ago, perhaps they're not so in mind. And the first thing I was going to talk about was what the viral load is. If I measure in your blood or in your mucus, if it's a respiratory disease, how much virus is floating around, then as a function of time, that what's called the viral load curve will have a particular shape, and that shape will be different in individuals. It may differ depending on whether you are immune compromised, whether you're young, whether you're old, whether you have diabetes, how much of the virus you were exposed to initially, whether you have a second virus, and so on. But for given individuals of a given health state, that curve is pretty typical in terms of its shape. And even between individuals who don't have identical health shape, those curves tend to be similar. Although, for example, lethal infections and non-lethal infections of the same virus may have slightly different curves. And there's this nice review paper by Amber Smith. Last year, we actually had Amber give a guest lecture. Maybe we can try to get her to give one this semester again. Would be nice. And she identifies a couple of different typical patterns of viral load. I should say that all of the y-axes here represent log scale. And so a relatively small change here actually is a 10-fold change in viral amount. It's not really indicated here. She has what's called monophasic curve, where the viral load comes up, comes to maximum, and then goes down fairly smoothly. A biphasic curve, where the viral load comes up, saturates, and then goes down very slowly, and then at some time switches to very rapid decay. A triphasic comes up, comes down, hangs up for some period of time, and then clears. And then the last one, which really shouldn't be named biphasic decay again, it's really persistence, is a situation where the virus never gets cleared. It's a persistent virus. HIV would come into that last category. We get a peak, your body then fights it, but never goes away completely. A lot of viruses, herpes are like that as well. There are other shapes as well, but one of your little homework assignments today will be, we're going to analyze flu, which has this biphasic curve, the second curve. I'd like you to see in the literature if you can find the viral load curve for some other disease and do the little analysis we'll do now for it and compare it. Does it fall into one of these categories or something else? And so on. If we look in vitro, so now in cell culture you don't have an immune system. And so you're not going to typically get clearance of the virus, because the virus is usually cleared by your adaptive immune system. But this is for COVID. If you put SARS-CoV-2 into a dish with cells that host SARS-CoV-2, you see nothing happens here for 6 hours. Then you have rapid exponential growth of the virus over the next 12 to 24 hours, well, actually 6 hours after that. So 6 hours to 12 hours, or 6 hours to 18 hours. Then there seems to be a slower growth of the amount of virus. And then starting here around 36 hours, the amount of virus starts going down. Again, this is a log scale, so going from 6 to 5 is a 10-fold decrease in the amount of virus. It may not look like a big change, but it's a pretty big change. The different curves here represent the amount of virus, a solid line inside the cells, dashed line in the fluid above the cells, because the cells start making virus inside themselves before they secrete it. And then the difference between the black curve and the green curve is that they're different cell types. And so the infectivity and the ability of the virus to reproduce in different cell types is different. So this is what you could do in vitro. It's easier to measure the amount of virus in vitro than it is in a human being or in an animal. And so you get more detailed data. If you're going to model viral replication, typically you have a model that looks something like this. This is again in that diagram from Amber's paper, although it's pretty typical of these kinds of diagrams in general. We have some virus in our environment, V. We have a susceptible cell, T. The virus and the susceptible cell combine to make an infected cell. It takes some amount of time for that infected cell to start producing virus to the outside world. That's called the eclipse phase, or the latent phase. Once that cell starts producing virus, it releases virus to the outside. At some point, that cell will die. That virus is cleared. It has a finite lifetime outside of the cells. And it can then go on to produce more infected cells. In principle, the uninfected cells can reproduce themselves. And so this looks very much like that birth-death SIR model that we had talked about briefly, skipped over a little bit last time. Has a lot of the same properties. And we're going to build one of those starting today. Okay. There are lots and lots and lots and lots of models of viral infection in the network form. I give you, as a reading assignment, paper of Erica and Jason Shoemaker on a couple of these. There's some other review articles that we mentioned. Sometimes these models are relatively simple, like this one. Sometimes, like this one of Erica's, they can be very complex. Depends on how much of the immune system you want to represent, whether you want to represent only one patch of tissue, or whether you'll distinguish, for example, the nose, the throat, the upper lungs, and the lower lungs as boxes in a model. And so there are all sorts of nice models of various kinds dealing with these things. And of course, depending on what virus you're talking about, influenza, HIV, this is another influenza one. This is a co-infection model. So this is looking at the interaction of influenza and streptococcus pneumonia. In this particular case, it winds up that the co-infection between the bacteria and the virus tends to make the viral infection much worse. There are other situations actually which being infected by one virus can protect you, because the first virus excites your immune response. And so if the second virus then comes in, it's not able to reproduce as much. There are other viruses where the first virus comes in, knocks the immune response down, and then the second virus could come in and do a lot of damage. So there are some interesting questions about that. So I want to come back now to that picture that we had from Amber's paper, where she had measured the viral load. This was measuring the viral load for flu, and it's measuring the viral load for flu in mice, not humans. And you'll see when you're talking about viral loads, you'll typically see this TCID50 number, and that's a sort of an odd looking unit to measure things in. And it's an operationally defined measurement. So the way I measure TCID50 is I take the fluid above the tissue and I put it into a cell culture, and I ask the question, and there are actually two different interpretations of this. One interpretation is how many cells does it kill in one dish? Another one of which is what fraction of the dishes that I in fact get killed. And so there's a little bit of a difference. But as you see, if I did it in an animal, it's what dosage I need to kill half of the animals I infect. If it's in a tissue, it's producing tissue damage and 50% of inoculated tissue culture cells. And so those might give slightly different numbers. The reason for this is a practical one. Until very recently, you couldn't actually see viruses. Still hard to see them. And so you can't directly count how many virus particles there are. You'd say, well, measure the density of the virus in the solution. Say it's X molar. Well, if you can't count them, you can't measure the molar concentration. The other problem with viruses is that you could have a virus particle sitting in your solution that doesn't have a complete genome inside of it. And that means that if you count it, you're counting it as a virus particle, but in fact, it never could go on to infect anybody. And viruses like influenza, which have rather complicated genome, where you have the RNA is packaged in 8 little separate pieces, a large fraction of influenza virions don't have complete genomes. And so those by themselves are unable to cause infection. And the estimates of what fraction of influenza particles are able to be infective, it varies, but it's typically on the order of 10%. And so this operational definition, which is the concentration of the virus is how much infecting that fluid can do, is practical. It's not an easy thing to measure because you have to do serial dilutions. Imagine you're going to measure this with animals. I have to take the supernatant of my virus out. I have to inject it into, I don't know, 10 animals, 20 animals, see what happens. And I dilute it by a factor of 2 and do the same thing again. And I keep doing that until I get to 50%. It's a very expensive and difficult thing to measure. And so surprisingly, knowing what the amount of virus in the supernatant is, it's actually pretty hard. And that's one of the reasons that when you see these data sets, you're going to see not a lot of data points. And in particular, the time intervals between the data points are often much longer than you'd like them to be. Another thing that you'll find when you look at these data sets is in humans, and I've mentioned this multiple times and I'll say it again, in diseases like influenza, you start feeling sick just about when the viral load is maximal. And so that means unless you happen to be sitting in the hospital for some other reason, and they happen to be sampling you to see how much influenza virus you had, you're not ever going to see this leading edge of this curve when the amount of virus is going up. Because by the time you're sick, the amount of virus has already gone to its maximum. And so if you look at human data on SARS-CoV-2, it almost always starts near its maximum. Because unless it was an experiment where you intentionally infected somebody, which they can't do with SARS, they do that sometimes with other viruses, you can't get that early time series because by the time a person's sick, you've already 7-8 days into the week's infection. And so there's a lot of missing information. With animal studies, you could do more, but because measuring the viral loads is such a heavy experiment to do, you'll find that the data are pretty sparse. And that's why I'm asking you to look at the literature and see what you can find. Okay. But if I look at this picture, amber is nicely labeled the regimes, 1, 2, 3, 4, 5. Maybe there should be a 6 here at the tail. This early phase was clearer perhaps in this picture here, where the TCID stayed effectively 0 for 6 hours. So there's this early phase when you put the virus into the system, the virus has entered cells, but it takes time to reproduce and come out again. So there's no new virus being produced. That could be called eclipse phase or lag phase. Depending on the virus, it could be anywhere from 15, 20 minutes to days. Then you have this rapid period of exponential growth, which you've got labeled as 2, when the cells are churning out a lot of virus and more cells are becoming infected and starting to churn out virus. And so you actually might expect to have a double exponential because the individual cells are exponentially increasing the amount of virus they're producing, but also the number of infected cells is going up. So you have a product of 2 increasing functions. And then you have this period 3 when that increase stops, and presumably you have the most virus in your system. You'll find for SARS-CoV-2, the shape is different. You see here, this is in vitro, not in vivo, but it goes up very fast and it keeps going up, but more slowly. And in humans, you'll see that too. You get this sort of more mountain-shaped curve rather than this very steep shelf, which is typical of influenza. Then you'll see in the case of influenza, this slow decline here in region 4. That slow decline, remember, is still a factor of 10 because this is a log scale plot. That's a big decrease. And then in the case of influenza, starting around day 7, there's a rapid decrease of virus. And if you're lucky, you clear it and the amount of virus goes to 0. If you're unlucky and it's persistent, then that level can stay non-zero, just the way in the population level, we never got rid of SARS-CoV-2. And so the peak of infection went down, but didn't go to 0. Now it's coming up again. And so it's possible also for viral infections. We were just talking to Erica and Bard to build models where you get a peak like this and then it goes down and then later on it peaks again. And there was a little bit of a homework problem asking you to explore that in the context of epidemiological models. So we'll see that, okay? So in the case of flu, there are actually region 1, there's a delay, region 2, exponential growth, saturation in region 3, slow exponential decay in region 4, fast exponential decay in region 5. And these different phases are in fact associated with different aspects of the virus and the immune system. And they're going to be quite different depending on the virus you look at. Any questions about that so far? So what we want to do is what we did by hand before, which is to get a rough idea of what the parameters are going to be that will constrain the models we build. Ultimately, we'll want to do model fitting, but as we saw from our epidemiology models, if we have a pretty good guess about the initial model parameters, we're much more likely to be able to actually use a fitting program to get us the results we care about. If you start very far away from the actual values, your fitting approaches typically don't work. And so we need to do their best to get estimates, rough estimates of these things. And so the first thing we're going to do look is region 1. Well, she really didn't sample the data very often. In this case, she sampled it every, let's see, basically once a day, if that. There is this little bit of a delay. It's hard to see in her model. It's actually, you have to really look in other data sets to get it in detail. But there's an initial lag from 0 to the beginning of this of about 6 hours. So influenza doesn't start reproducing inside cells for 4 to 6 hours after somebody coughs on you. You've got 4 or 6 hours where your cells aren't producing flu. So we'll say 6 hours. You could argue that. Okay. The next thing that we have is starting at 6 hours, going up to a day. We have this exponential growth. And again, we only have 2 points, so we don't really know what the shape is. But we know that the increase is big. And we've gone from 0 here at the end of our day. We're extrapolating. We're up here. We're around 6. And so over one day, our TCID has gone from 0 to 6. That is, if that's on a log scale, so it's gone from 1 to 1,000,000. So you've had a 1,000,000-fold increase over 24 hours. That corresponds to a 10-fold increase over 4 hours, or a 10-fold increase over 1/6 of a day. So the time to increase by a factor of 10 is 1/6 if we're measuring units in days. Is that okay? We're going to make all of our measurements in units of days. Okay. And then we need to know what the level was when we saturated. And if we just look at our picture here, it's about 6, which is 10 to the 6th, about 1,000,000. So if our initial load was 1, after a day, it's 1,000,000. So viruses are pretty fast and pretty efficient at getting your cells to turn them out. So increasing in concentration by 1,000,000-fold over a day is pretty impressive. I don't think we have many industrial processes that can do that. So we get saturation of 10 to the 6th, and we said we started around 6 hours and we're ending around a day plus 6 hours, more or less. Okay, so that was our region 3. And now we have this time period of relatively slow decrease. We start around 6, we wind up around 5, and the interval is between 2 and 7. So we have a 10-fold increase over 5 days. So Our increase was tenfold over a sixth of a day. We're now decreasing tenfold over 5 days. And so in one case, our time constant was 1/6. Now our time constant is 5. Okay. And then in our region 5, which starts around day 7, our concentration goes down from 10 to the 5th, back down to 0 to 1. So now we have a 10 to the 5th decrease from day 7 to about day 8. And that corresponds to a tenfold decrease over about 2/5 of a day. And you could argue about the details, but now we have some rates. So we have eclipse phase. Some of these are rates and some of these are times, so we should probably be a little bit more careful. Our eclipse phase lasts 6 hours about, so the rate is 1/6 of an hour. If we do it in days, 6 hours is a quarter of a day. So that would give us 1 per 4 days. I mean, sorry, 1/4 of a day. The exponential growth phase is tenfold over 4 hours. Again, so 1/6 of a day. The saturation is at 10 to the 6th. Slow clearance is tenfold over 5 days. Rapid clearance is 10 to the 5th over 2 days. And so again, then we get a factor of 10 over 2/5 of a day. So if we're going to do everything by tens, then we have to be consistent. And we'll need those numbers going forward, so it's important to do that. And so what I'd like you to do in your homework exercise is find a viral load curve, ideally one where they measure more frequently than this, but at least as frequently as this. Look at the shape, and you'll have to say, depending on the shape you find, you'll have to say what the regimes are. This one, as I say, we defined 5 regimes. Some viruses might have more regimes or fewer. And then within each subdomain of the regime, you have to say when it starts, when it ends, the fold change in the values to get the rates, if you're going to fit it. Okay. Okay, so now we're going to try to build a little model of viral infection. The one we're going to build is one I did over the summer. It's not a particularly elegant or beautiful model of viral infection, and I think people have already run into some nicer ones than the ones I'm going to show you. But it's the one that we have worked up, and so you can definitely use it as a framework for building a better model. And so as usual, if we're going to try to reproduce this curve that we've seen, in reality, the viral load by itself doesn't constrain your model very much. And so we'll find we could write many different models that would give us the same influenza viral load curve. So in cases where there is additional information, you're stronger. So if somebody's experimentally measured the number of CD8 plus T cells, which are one of the key components of the adaptive immune system, then if you have the viral load plus the CD8 T cell load, that helps you a lot in terms of constraining your model. If somebody has measured the level of an interferon or other cytokine, an interleukin, that can help you constrain the model a lot. If somebody's measured the amount of cells that have died, the number of cells that have died in your lungs as a function of time, which people have done for SARS, that can be very helpful. You can imagine other kinds of measurements that could be made too. There are more than one immune cell type that could be measured. Recovery of the tissue after damage is possible. Multiple chemical signals related to inflammation and suppression of inflammation. Temperature, if you have a fever, fever is associated with interleukin 6, also other things, but interleukin 6. So if you measure the body temperature as a function of time, that can be informative. Some of these viruses affect the way you're able to breathe. So your blood oxygen level could be a monitor of how sick you got, how much lung damage you have. That could be good. In the case of mice, when mice get sick, mice have very high metabolic rate. You starve a mouse for a day, it more or less dies of starvation. And so they actually lose body mass appreciably. If you don't feed a mouse for a day, its mass goes down because it has such a high metabolic rate. And so in the case of mice, the body weight of the mouse actually reflects how sick it is pretty well, maybe with a 12-hour delay. And so if you put your mouse on a scale and it's losing weight, it's sick. And Amber has shown in her experiments that loss of weight of a mouse is a very good surrogate for the area of damage in the lungs, which is much harder to measure. In the case of monkeys, it doesn't work. If you put a monkey on a scale, it can be dying of influenza and it still doesn't lose a lot of weight. Basically, the bigger the animal, the more reserves they have, and so the less weight reflects illness. Unless the illness is a slow one. If you have cancer, HIV, then of course weight loss later on in the process of disease is typical, but not so much early on. Mice have rapid reported. So that comes back to some of the things Kali was talking about lags. If you wanted to use weight as a reporter, the delay between the time you're sick at a given level and the time you lose weight would vary depending on the species. And so having a lag there would be quite important. Okay. So we're going to begin by going back to the very, very simple model that we started with, which is just thinking about infection and viral reproduction. And I'll show the slides that we did back in lecture 1, just to remind you, and we'll then move in further. So if we think about the biology of viral replication, it's actually pretty complicated. There are a lot of steps between the time a virus touches the surface of a cell and the time new virus is released by the cell. A lot of things have to happen. Almost all of the models we'll look at are going to neglect almost all of that complexity. On the other hand, if we were trying to develop an antiviral drug, we'd have to think about that complexity in detail. And that complexity may actually have some interesting and significant effects on how these things happen. But most of these models make do without it. And some of those models are very clinically successful. So that's a simplification that seems to be, if you looked at the biology, you'd say you have to include all of these things. You look at the models, you don't include any of them, and they still work. So sometimes you can be surprised. So what has to happen to be infected? First, the virus particle has to come in contact, usually with a specific receptor. In the case of SARS-CoV-2, that receptor would be ACE2. There's a co-receptor, TMPRSS2. You don't need TMPRSS2 to be infected, but the infection is faster. There are two different modes of entry of the RNA. In the case of SARS, we're not going to model those separately. Flu is different. But in either case, the net result to become infected is that the virus capsule has to be dissolved and the RNA, which is the viral genome, has to be released into the cytoplasm. In the case of influenza, there are actually 8 separate viral RNAs that have to be released into the cytoplasm. Those viral RNAs have to find their way to a ribosome. And that ribosome has to transcribe those RNAs into protein. And those viral proteins then have to do things. In particular, one of the first proteins that they're going to make is RNA-dependent RNA polymerase, which is a viral protein that copies the RNA. Because in your body, in general, there are no molecules that copy RNAs. So if you're an RNA virus and you want to replicate yourself, you have to make your own replication machinery. And that viral-dependent RNA polymerase, is shown here, will then make copies of the viral RNA. Some of those copies of the viral RNA are then broken in pieces and sent back to ribosomes, which make viral proteins, additional viral proteins. That first viral protein has to be makeable without destroying the RNA, because if it did, then there wouldn't be any RNA to copy. And they typically come off the minus strand rather than the positive strand. And so now you have one set of machinery, which is the RNA-dependent RNA polymerase, which is making copies of the viral genome, and a second set of machinery, which is host cell machinery, which is using the ribosomes to make the viral proteins. Somehow, the RNA, the viral RNA and the viral proteins have to come together and assemble to make a new viral particle. And exactly how that works depends a lot on the virus, and it's rather mysterious even when we do know what happens, exactly how that works. The self-assembly is quite complicated. There are people like Adam Zlotnick at IU who specialize in studying that. And then the viral particle, once it's been assembled, has to get out of the cell. In the case of SARS-CoV-2, it uses the normal secretory mechanisms that the cell uses to let things out, so exocytosis. That means that making the virus direct doesn't automatically kill the cell. There are other viruses called lytic viruses where essentially the amount of virus just builds up in the cell until the cell bursts. And so no virus is released until the cell dies. In this case, it's not like that. And then the last thing is that the virus has to get out of the cell, but it winds up, they tend to be sticky and stay stuck to the cell membrane or to proteins in the cell membrane. And so often there has to be a special protein that cleaves the virus and releases it into the environment. And any of these steps can go wrong for the virus, which is why you wind up with so many dud viral particles. And in electron micrographs of SARS-CoV-2 infected cells, you see that it looks like that release is not very effective in a lot of cases. And so in the electron micrographs, you'll see a few viral particles that have released and lots of viral particles that seem to have just stayed stuck to the surface of the cell. They come up to the surface of the cell, they stay stuck there, and they can't go on to infect other cells. Another reason that just counting the virus doesn't tell you what that TCID number is. And so now we're going to come back to the model. Our model is going to neglect all of that complexity. And we're going to come back to our classic SIR type model. We're going to assume that there's some amount of virus in the environment. There's some population T, which corresponds to our old S, which is cells that are uninfected. Those T infectable cells, those target cells, come in contact with the virus. When they do, there's some probability, beta, which we remember from SIR models, that our target uninfected cells becomes infected. And we're calling it I1 because we're going to assume that there's this lag time between the time of infection and the time the cell starts producing new virus. Okay. And there's already an issue, which is that classically when people write this equation, they say the rate of transition from uninfected to infected is beta times T times V. We've seen that before. Our terms should be normalized by the number of cells, which is not true here. And also, in this case, when the virus enters the cell, it's no longer outside. And so the same viral particle can only infect one cell. The viral particle is destroyed when it infects a cell. It's dissolved. And therefore, in principle, we should be writing this in the form of a chemical reaction. T plus V goes to I1, rather than in the form of regulation, T goes to I1. What's the difference? In this case, the virus is used up when it infects the cell. In the other case, it's not. And one thing we could explore is whether that makes a difference. It's going to tend to make a difference when the amount of virus is small. If you have 5 virus particles in the environment, then at most they can infect 5 cells because they're used up. If you assume that they're not used up, in principle, those 5 virus particles could go on and infect an infinite number of cells. And so you're going to get a huge difference. If the number of virus particles that are in the environment is big compared to the number being used up, then that approximation is not significant. Almost all of the models I have seen assume virus is not used up on infection. My read of that is that in general, the numbers work so that it's not significant, but it would be worth checking. Okay. Okay. And so this comes back to my comment, something that we've talked about a little bit, but really it came up in the rate of infection of susceptibles also in the population, where we had to normalize by S0, the total population. The same thing is coming about here. If I look at this equation that I've written here, T goes to I1 beta times T times V. Mathematically, that's a well-formed rate law. And if you're a chemist, it looks like what's called a mass action term, which is what you expect. But if you think biologically, that's actually not a meaningful term, because V is the total amount of virus in the system. And an individual cell doesn't know how much virus is in the system. It only knows how much virus is locally visible to it. And so you should always write something like a rate of infection in terms of something that's biologically meaningful, in this case, the amount of virus per cell, and not the total amount of virus. If I make my system twice as big by copying it, I have 2 copies of it, I'm not changing the rate of infection of individual cells. And therefore, the size of the system should scale out. And so you really should write this beta times T0, total number of cells, times T times V divided by T0. Of course, the T0s cancel each other out. But the real rate law should be beta times T0, so a much bigger number, because T0 will be number 10 to the 7th, 10 to the 6th, 10 to the 7th, times T, that's okay. And then the amount of virus per cell, which is much smaller. And so that's something that you want, when you're reading papers, you better watch out for that, because a lot of people are sloppy about that. And very often it means they come up with results that make no sense. So the moment you see this equation, beta TV, without that kind of normalization, be skeptical. Don't reject the results outright, but be cautious. Okay. If the initial number of susceptibles is T0, we start out with no infected cells, then in this case, since the amount of virus does not depend on the anything, it's a constant, the number of susceptibles just is their initial number, e to the minus beta VT. So we get exponential decay. And that was the very first exercise we did to introduce delirium back a month ago. And we did the simple simulation of that. And I'm not going to ask you to do it again now, but if you felt like it, you could do it again. And we saw exponential decay. So now we're going to begin to think about what happens next. So once we have those infected cells, they're early infected cells, they have the virus inside of them, that virus is going to start replicating. And sometime after it started replicating, it'll assemble itself, go through that whole long viral replication cycle inside the cell, which is pretty complicated. And then whether it's a lytic or a non-lytic virus, at some point, if there are going to be more cells infected, the virus has to get out of the cell. And there's some amount of time that the virus takes to get out of the cell. Now, in reality, probably there's a period of time when there is no virus getting out of the cells at all. And so there's truly a delay, a lag. People tend to neglect the lag and talk about a rate at which you go from early infected, which is having the virus replicating inside of you, to late infected when you're starting to produce virus. And whether it's legitimate to represent a delay by a rate is an interesting question. Kali alluded to it. But for the moment, we're going to assume that we can do that. And so we're going to say our I1 cells are cells that have been infected but are not yet producing virus. And our I2 cells are cells that are infected and are producing virus. And based on the estimates that we did before, we said that this takes around 6 hours. That is a quarter of a day. And that means the rate, K, the rate of transition is the time 1 over the time. So if the amount of time it takes is a quarter of a day, then the rate K would be 4 per day. Is that okay? So it's usually easier to think about delays rather than rates, but the parameters we're going to use are all going to be rates. So we have to get used to the process in our heads of saying a delay of a day means a rate of once per day. A delay of 2 days means a rate of 1/2 per day. A rate of a delay of 6 hours means 4 per day. Okay. And so we're going to now assume that if our eclipse time was 6 hours, a quarter of a day, that means that the transition rate, 1 over that, is 4 per day. And so we write a term which is di1 dt beta TV, that's our rate of newly infected cells. Our I1s become I2s at a rate ki1. So we have beta TV minus ki1. And we have a balanced term di2 dt equals ki1. We add these together, the total number I1 plus I2 is just beta TV, rate of new infected cells. For SARS-CoV-2, that lag is about twice as big. It's about 12 hours rather than 4 hours. And so it's about 2 per day rather than 4 per day. That's why measuring these things by hand is useful. So we have a rough idea of what's going on. Okay. So let's actually do this. Some of these slides come from the lectures I gave over the summer when we were introducing antimony. And so occasionally you'll see some things that are very elementary that you know already. I've tried to clean them up, but I haven't always gotten all of them out. So there's a comment in antimony, we specify ODs as transition rules. Well, you'll remember that. So what I'd like you to do now as our first exercise, and this is something that we can do together, but I'll do it as an exercise, is I want you to write the model as follows. T goes to I1 with a rate beta times T times V. Notice V is a constant now. Beta is a constant, T is T. And then I1 goes to I2 at a rate K I1. And we'll use, for the beginning, a beta is 6.2 times 10 to the minus 5 TCID times cells times day. And this is going to wind up being important, dependent on the initial population. And the initial population that was used was, the initial population that was used was 10 to the 7th cells. So my real beta would be this number times that. So it would be 6.2 10 to the minus 5 times 10 to the 7th. So it would actually be 6.2 times 10 to the 2. And then the amount of virus per cell would be whatever it is here. And our initial V was here was 10,000. We'll fix that in a minute. And that would then be divided by 10 to the 7th. So we have here 10 to the 4 divided by 10 to the 7th, 10 to the minus 3. It's the amount of virus per cell. So one viral particle for every 10 to the 3 cells. Then if you do it that way, then changing T naught doesn't change it. Okay, is it clear what the little exercise is? So I want you to write a simple simulation. I've got it actually here written out, so you could copy it, but I'd rather you try to do it on your own. T goes to I1, I1 goes to I2, and we can pull the parameters beta, 6.2 10 to the minus 5, K, the rate of conversion, 4 per day, T0, 1 e to the 7th, and initial virus, 1 e to the 4. Okay, so that's our little assignment. So I'll let everybody launch Tellurium and try to get that run. And I'll do my usual thing, which is I'll set the poll up and just don't fill it out until you're done. When you've finished it, answer that you've finished the exercise. So that way you don't have to redo the poll, but it's there so I can keep track of how people are doing. Everybody okay with this as an exercise? Any questions about that? Good, okay. So take a few minutes and give that a try. And if you got it very quickly, why don't you write a little parameter scan that changes the value of K, changes the rate at which, that's how long it takes before the virus is produced, and overlays your plot so you can see what happens as you change that time. You could try, for example, the delay is 3 hours, 6 hours, 12 hours, 24 hours. What happens? So if you got it done faster, then you have a little extra assignment. Okay, I think there's still one person working on it. We're at our 6:30 mark, so why don't we take a 5-minute break? The person who's still working on it will continue to work on it for a few minutes. And then if you need to take a break and need a little longer for the break after that, that's fine. But why don't we break now and plan to start back at, say, 6:37, if that's okay, and then we'll continue then. Okay, is everybody back? Anybody need more time before we get started? Okay, great. Okay, so this is what I got from a while ago. What I was changing here in this simulation was that delay time. In one case, I had an eclipse phase of 6 hours. And in the second case, I had an eclipse phase of 12 hours. I see all my cells get infected. The little bump is this I1 phase, that early infected phase. And I see that in the case of the longer delay, that I1 phase lasts longer, so I get more of them. But it doesn't really change anything else. Everybody is infected pretty much by 10 hours. I mean, sorry, 10 days. I keep saying hours, so please correct me. 10 days. The duration of the infection is a little bit slowed out. And if you look in detail down at the very beginning here with this green curve, you'll see the green curve isn't going up at the very beginning. It's got a little bit of a concave region to it. And that concave region is a little bit bigger in the case of the 12-hour delay than in the case of the 6-hour delay. If I made the second delay 24 hours, so a full day, or let's make it 48 hours so that it's really a bigger difference. Let's try that, see what it does. Yeah. So if I make it a 2-day delay instead of a 6-hour delay, I have quite a big difference. The amount of early infection I get is longer because I still stay early infected longer. And I see that there is a substantial delay. The thing is still pretty much over by 10 days, but at 6 days, I still have a substantial fraction of uninfected cells in this case and not in the other. And so that delay does make a difference, especially to the early behavior and to the total number of infected cells early on. It's not going to be so significant here, but once we have the cells actually producing virus, and the amount of virus is no longer a constant, but depends on the viral production, then you'll see something quite different. Okay. Did anybody have any different results from that when they scanned that K parameter? I think that's the main concept. Okay. So if we want to make this model more interesting, we have to introduce the concept of the cells producing virus. And these late infected cells will produce virus. And since we don't have individual cells, we don't really have an ability to model that some cells might produce a lot of virus, some cells might produce a little virus, or the fact that the amount of virus produced by an infected cell may not be constant in time. In general, in the case of SARS-CoV-2, the cells which start producing virus initially produce a small amount, and then as the RNA is replicated inside the cell, the amount they produce per unit time increases exponentially until it saturates the synthesis machinery of the cell. And so, in fact, to assume that infected I2 cells produce a constant amount of virus per time is a pretty poor approximation. Again, somewhat surprisingly, we're going to find that that is how people model it, and that those models work pretty well. So that all the detail of how the viral replication works doesn't seem to be that critical for many of these things. And the key thing we're going to have to remember here is the rate of production of virus, because ultimately it's that amount of virus in the extracellular space, that V term, that we're going to be calibrating against the experimental measurements. And so this quantity now is going to be a critical one in our model. And so we're going to assume that infected cells produce virus at a constant rate, or that rate will be called P here. And so the amount of virus in the extracellular environment, dV dt, will be P times the number of late infected cells, P times I2. And the units of that are in TCID, that funny unit that we've used to measure extracellular virus concentration, divided by the number of cells, divided by days. So again, the rate constant sometimes have units that are a little bit funny, or at least a little bit not intuitive, but it's important to always remember what the units are. We should never write down amounts without units. And in fact, in this particular example, I've been sloppy because some of these things do have units. For example, beta has units of per TCID per day. K has units of per day. That's good. These should have, say, numbers of cells. This should be in TCIDs here. So we have to think about that a little bit. So now we're going to add the virus production. Now this is now going to change our model fundamentally, because in this base model, V was a constant. Now V is going to be a variable. And so now we have a loop where the virus infects our susceptible cells. They go to I1. After some time, they go to I2. They start producing virus. And so the rate at which they produce is something we don't really know. And we're going to just arbitrarily start out by saying they produce 1 TCID per cell per day. And that's a number that we're going to have to adjust. I'll point out that what we really care about is the product of P and beta. If we make beta 10 times bigger, we can make P 10 times smaller. It doesn't change anything. And so I'm going to ask you now to do that as a little exercise. So I'd like you to take the model that we have here and add this P I2 goes to V term to it. And see what happens. And this is actually now going to look, what we had here looks like an SIR model. When we add virus, external virus, we're going to get something for the first time that's different from what we've done so far. And so I'll let everybody, I'll do the same thing with the poll. I'll let everybody add that. Take your time trying to do that. And I'll say that this level of model is actually something that people use. You'll see published papers that don't have models that are much more complicated than this. And I guess it's worth saying that the production of virus is shown here as it would show up in antimony. Nothing goes to virus because your I2 cells are not used up in the production of virus. So it's a regulatory reaction rather than a chemical reaction. Okay, so everybody seems to have gotten that to work. Let me see if I can get it to work. E3 colon, nothing goes to V at a rate T times I2. And then we said KP was equal to 1.0. And the units are TCID per cell per day. And what I got was that. Did people get something that looks like that?
Yep.
So the first thing to notice about that is that plotting the amount of virus on the same plot as the amount of cells is a bad idea. Because the amount of virus is increasing essentially exponentially. Actually, well, linearly in the first part, exponentially the second one. And so it's not on the same scale. And so probably if I'm going to do this right, I should put them on separate plots. And in this particular little example that I did here, I put them on separate plots. And you'll notice that the shape of the curves is quite different from what it was before. And perhaps it would be nice to have the old, I'll go back so we could look at the old figure. Look at the difference. This is actually now over a much shorter time scale. I've put it over 2.5 days instead of 10 days. But look at the difference in shape. Instead of having the number of uninfected cells decrease exponentially from the beginning, which is what happened before, the number of uninfected cells initially is constant, because until I start producing new virus, I don't get any new infection. I get a few cells infected at the beginning, and then I don't have any new infection until my infected cells become I2s. And so in the case of the original model, the rate of decrease of the rate of infection was maximal at time 0. In the case of the new model, the rate of infection is nearly 0 at time 0. So that's a huge difference in response. And then instead of having this smooth exponential decay of my susceptible population, what I see is a shelf followed by a very rapid decay, followed basically by a stop. All of my cells have become infected by here a day and a quarter. The shape of my I1 population here is different too. On the leading edge, that increases fastest at time 0. Here, it doesn't increase at all at time 0. And the shape of my bump is different. It's concave on both sides. And this one, it's convex on the left-hand side and concave on the right-hand side. And so the short time behavior of these models is totally different. Now, if I had plotted the amount of virus in my old model, it was just constant, nothing happened. In my new model, the amount of virus starts out as 10, whatever it was, small. It increases exponentially, which it's supposed to do, and then it saturates. And there's a delay before it starts increasing. And so this model is already beginning to reproduce the beginning of my viral replication curve, my viral load curve. I've matched the delay. In this case, I've set it to be a quarter of a day, 6 hours. And I have my exponential growth phase. And in this curve here, I was around 10 to the 6th at day 1. And if I go over here, I'm at 10 to the 6th at day 1. And so it happens that the release of 1 TCID per day and that delay of 6 hours, a quarter of a day, gives me the right shape for the beginning of the experiment for influenza. Now, I could have picked a different number for P and then it wouldn't have agreed. And if it didn't agree, then I'd have to adjust it. So what would happen here? Suppose I made my P value 2 DCIDs per day. Well, it doesn't change my delay very much. It means my saturation value is a little bit higher. It's about 310 to the 6th instead of 10 to the 6th. It actually doesn't make a huge difference. So the p-value didn't make a huge difference. What if I made it smaller rather than bigger? I started out with 1. Let's make it 0. Let's make it a 10th as big. What do you think is going to happen? Why don't you try it? Make it a 10th as big. What happens? Does it change anything? What do you expect? If I make the amount of virus produced 10 times less per cell, what's going to happen? Anybody? Slows everything down. What if I run for longer? Does it actually change the total amount of infection or just slow things down?
Should just slow things down, right?
Yep, just slows things down. I went maybe, oops, I don't know what I did there. Oh, I typed an extra 2. So it slows things down. My peak of I1s is a little bit lower. And here, the time to get to my maximum is from, let's see, when do I get my inflection point? About 2.5 days. So it slows things down by about a factor of 2. Okay. So actually, this very, very simple model is already reproducing the first 2 phases of our virus. That's not bad for a model with 3 terms. And that explains to some extent why people like these very simple models. They could actually reproduce these viral load curves pretty well. At least the flu viral load curve. For some diseases, the viral load curves are very complicated, and then you may not be able to reproduce it so. Okay. So I want to come back to this idea, and I should probably try to rescale these things so that they're all consistent in terms of fold changes and hours instead of days, instead of having them in different units. The eclipse phase is a quarter of a day. Exponential growth phase, 10-fold over a 1/6 of a day. Saturation phase, 10-fold over a 1/6 of a day. Clearance, 10-fold over 5 days. Rapid clearance, 10-fold over 2/5 of a day. So I should really, Josh, if you could make a note to redo these tables to be consistent that way, that would be good. So it should always be 10-fold and then the time. Okay. So that's where we had it so far. And now let's think about how the parameters affect things. We've already changed here one of the parameters, which was the rate of viral production. I'll put it back to 1. Now I have to change my time which I plot too. Why don't you try changing beta now, the infectivity, and see what happens. If you have more, if the virus is more infectious, you expect the sweep of the virus to be faster or slower. If the rate at which the virus infects cells is faster, do you expect the virus to progress faster or slower?
Faster.
Faster. It's not a trick question. It's such a simple question. People are thinking, is there a trick hiding there? It wasn't a trick question. It was just a basic question. So we could try changing that. If we change the eclipse phase, we already saw in the base model that the longer the eclipse phase is, the slower things happen. So why don't we all try that together? We could make if we make the delay bigger, slows things down. That's not surprising. If we make our infectivity bigger, that didn't seem to do much, but I didn't compare them. Let's see. That has a peak around a day and a half. Let me make it tenfold bigger so it's more obvious. It speeds things up a lot. So it seems to behave as expected. If you make a real parameter sweep, then you could see this more consistently. I'm just going through my hand and doing it. So why don't people just spend a few minutes playing with that and exploring those 3 parameters to get a sense of it. And I'll play with it for a few minutes and let me know when you've had enough time exploring. That number 4, try changing the infection rule to T plus V goes to I1, is interesting, and it's not going to work. And the reason it's not going to work is that this issue about normalization of the rate loss, that winds up being important in that case. But it's worth trying it. You'll find that if you wanted to get something that looks like the correct curves, when you actually have the virus used up, you have to change the parameters a lot. And since the base model we're working from doesn't do that, we're not going to spend the time on it. Although that would be an interesting exercise to do as a homework. But let's focus on the first 3 of these first, and then we'll come back and try that one. where we have the combination one together. I think you'll find as you do this, you can have quite in the basic shape, which is going to be similar. If you look here, where I slowed down the K a lot, so that the conversion from early infected to late infected is very slow, I stretch out the I1. I2 is delayed, and the virus increases. Actually, not as the change in viral load isn't as big as you might expect. So let me see if I can undo what I just did to put things back the way they were. I'm back to our base model here. Yeah. That was our base model. Did anybody try changing the infection rule? That's easy to do in terms of writing it. It would be T plus V. What happened? Did it change things a little bit, a lot? What did you get?
I personally got errors.
Errors. Well, maybe see what happens. Okay, well, if I do it, nothing happens. Everything goes away. Why is that? Why does nothing happen? And I could make these times longer. Let me make these times longer. Add some data points so I don't have convergence issues. Well, maybe not so different. Everything gets slowed down. But ultimately, I see the same kind of curve, it just is delayed a lot. The viral load curve has a very interesting shape, though, very different. Why do you think that could be? Well, what's happening to the amount of virus early on? Suppose that I start out with, here actually, I'm sorry, this model that I'm running started with early infected cells and not with virus. Let me start with virus and no early infected cells. So I actually pulled a switch on. There we go. So I'm now going to start with viral load and amount of virus here, 10 to the 5th, which is high level, high TCID. What's happening? Let me go back to that faster time scale. Need to make this about 7.5. And let's see what's happening. The viral load curve is a lot more complicated than it was before. Somebody tell me why we're getting this shape. Any ideas about that? So what's happening at short times to the virus, amount of virus in the system? Is it increasing or decreasing?
I can't tell if it's increasing and then decreasing really quickly or if it's just decreasing.
Okay, well, that's a good point. So let's zoom in on that. Let's look at, and you guys can always be doing the same things. Okay, so it's decreasing a lot, and then it starts coming back up. Why is that? Well, initially I have 10 to the 7th infectable T cells and 10 to the 5 virus. So if they all combine together, how much virus do I have left after they combine?
None.
None. So with this kind of law, if beta is reasonably big, all the virus immediately disappears into the cell. There isn't any circulating virus anymore. And then I have to wait for my I2 cells to come and start producing virus. So if beta were smaller, that combination would happen more slowly. See, that slows things down. And the reason I want to walk you through this is that when I ask you a question like, why does something happen or what happens, it's not easy to understand that to begin with. And I want to walk you through a little bit of how one might think about analyzing this problem. And give you some insight into, if there were a homework question, we're not probably going to have a test, but if it were a test question, what would it look like? Or if it were a problem where you didn't know what the answer was to begin with, what would you do? And so these are some things one might think about in terms of analyzing this problem. So the viral load goes to 0 because all the virus is used up. If I started with less virus, it goes to 0. And in this case, because the total number of infected cells is very, very small, it's going to be a very long time indeed before I have enough cells producing virus to make any difference. But still, eventually it comes back. Now, what did I change? I changed the amount of virus. Okay, and then put this back to 10. Oops. Okay, so now we understand why it goes down. And it starts coming back up when we have our I2 cells producing virus. Why does this curve have the shape it does? It's a rather complex squiggly shape, isn't it? Tell me what's going on in this region here between 1 day and 5 days. It's pretty flat, right? My amount of virus is increasing in a straight line here. That means it's going up exponentially. Why is it going up exponentially? What's controlling the rate of production of virus? Somebody
Is it-
want to guess?
The P
Because it's
variable.
E or I2.
P times I2, right? So if this is increasing, the slope, remember this is exponential. So a straight line here means that the slope keeps going up. It's a log scale. That means that the number of infected cells keeps going up. If I look at my I2s, they're increasing exponentially here. All right. Why does the slope get steeper around day 5? Why am I getting more virus suddenly at day 5? The rate of increase of virus in my system goes way up. Why is that?
Well, at day 5, it looks like that's where the curves for the number of susceptible and number of late stage cells are equal. So then you have the late stage cells releasing viruses that are going to go and infect the susceptible cells. And if they're like in equal numbers, then every single susceptible cell should probably get infected. So that's why there's a big increase.
Right. Well, the only thing that takes virus out of the system, the way we've written it, in reality, the virus would decay. We should include that. But because it doesn't last forever in the environment, actually only lasts about a few hours before it decays. But the only way virus is taken out of the system is by being eaten by our susceptible cells. Around day 5 to day 6, the number of T cells is very small. And so that means that the rate at which the virus is being taken out is disappearing. So fundamentally, this plus V, which is taking virus out of our system, turns off. So essentially what's happening here is this curve here is what I would have gotten if I hadn't had the virus taken out of the system in the first place. If I'd written just T goes to I1 like that, that's the slope I would get. Does that make sense? I can try to explain that in different words if it doesn't make sense, but I hope that's clear. All right, so what's happening here is that this is when the cells are producing virus, but a lot of the virus is being used up infecting new cells. Here, the cells are producing virus, and there are no new cells to infect, so this virus isn't getting used up. Why does it flatten out here? Why doesn't it keep going up at this fast rate? It's not because I'm taking virus out of the system. What's happened?
Is it because most of the cells or all of them have entered like the I2 category?
Mm-hmm. In this little region here, between around 5 days, I have a lot of cells switching from I1 to I2. So I'm adding cells to the production. After that, the number of I2 cells is fixed. And so I just produce I2 times constant, maximum I2 times P virus. And so this slope is constant. This rapid region is associated with the disappearance of my I1s. And so if I slow down that transition between I1s and I2, it should stretch things out. So if I do that, notice that I1s last longer. I get a bigger I1 peak. The transition to I2 is slower. And that region of rapid increase is flattened out. If I slow my K down even more, stretches things out more and slows things down even a little bit. So on the one hand, this curve for viral load looks very complicated. It goes down very fast, then it comes up very fast. Then there's this exponential increase with a constant exponent for a while. Then there's this fast exponential increase, then it slows down, and then there's a slow exponential. That looks very complicated. But we can actually look at what's happening to our cell population and look at what our equations are doing and the dependencies and actually understand. And I understand that doing that automatically may take some time. We can actually understand what's going on biologically by looking at this picture, interpreting it through the lens of the equations we've written. If we didn't have these equations and we just saw this very complicated curve, we'd have no idea what it meant. But if we believe that this set of equations generated this curve, we can actually understand why the curve has the shape it does. And that's to some extent the value of the mathematics. The experimentalists give you some crazy viral load curve, and they say, we don't know why it's got this weird shape. Why does it go down and then up and then down and slow down and speed up and slow down again? And you can say, well, even though these equations look very, very simple, there are these different regimes. In one regime, the rate of susceptible cells is decreasing fast, then it stops. The rate of increase of our I2 cells is fast, and then it stops. That defines the transitions between the two regimes. We could probably play around with this and come up with some different shapes, but I'm not, and that would be an interesting exercise to do as a homework assignment, for example. But let's see if I change. Here, basically the only rates that matter are beta and K. I don't think I can do very much else beyond that. Let's try slowing K down, stretches everything out, doesn't do much else. So that's about all this model can do. Give us the shape. Now it happens that this shape is not a very good match to what we see experimentally. And so that's perhaps why people don't use this combination model when they're doing their modeling. In reality, that combination is happening. And so it's a bit mysterious why putting it in makes the model worse. And the answer is that in reality, we're leaving out a lot of other things that are important. Many questions about that so far. That's actually quite a lot of biology in a simple model. This slide somehow got misformatted, and I apologize for that. The next thing that I wanted to do, and it was an alternative to doing this combination, is to include the fact, which is what I mentioned, that the virus doesn't stick around forever. And that's an awfully good thing, because if it did, none of the antiviral mechanisms that we're working on, like social distancing and masks and everything else, would work. Because if the virus stayed in the air all the time, so that if somebody had walked by 2 hours ago and you walked through and you got infected, then life would be even harder. And early on in this infection, in this disease, there were lots of studies about how long the virus could survive on a surface. people were worried that when they ordered out from a restaurant, on the styrofoam container, there could be live virus. Well, there could be. You could get infected from that. And it's certainly possible, for example, doorknobs are classic ways of spreading. Classic SARS was spread especially by contact. And this SARS may be too.
And those are one of the cases where doing TCID is important, not just in the context of within host viral kinetics, but just to understand the stability of infectious viruses, because most of these studies are done by running qPCR and then detecting viral genome. But viral genome can lay on surfaces much longer than the actual infectious virus can remain on the surface.
Yeah, that's important. That to infect your cells, the virus has to have a viral capsule that's correct, and it also has to have the RNA inside of it. The capsid minus the RNA is not infectious, although it'll be taken into the cell, but it can't create illness because the genome isn't there. The RNA by itself in general can't get into the cell by itself. It needs the virus capsid proteins to be able to get in. Again, I suppose there's some minute possibility that the RNA directly could get into the cell, but that's very, very low. And your skin in particular is full of RNases. Your skin is full of molecules that break down RNA, precisely because there's a lot of viral RNA floating around. And if you ever, I don't know if any of you have ever done lab experiments in a biology lab, but if you're working with RNA, you have to be super careful not to touch anything, because if you touch a test tube or a pipette or anything, the RNases in your sweat are extremely effective. And so the RNA that you're trying to process will get broken down in an instant, not an instant, but really fast, by even a tiny amount of sweat from your skin. If the RNA is inside a viral capsid, it's protected. If it's loose, it's not. And so Josh's point is a very good one, that on a surface, the RNA may persist for a long time, but if the capsid isn't there, that's not infectious. But that's going to vary depending on the virus and depending on the details. There are some viruses that are primarily transmitted over surfaces, some in the air. In addition, if we're talking about the virus in your lungs, for example, or in your nose, we're talking about not the lifetime on a surface like my desk or your cell phone, but rather in your lungs. And there are additional clearance processes. In your lungs, the mucus in your lungs is continually being pumped up out of your lungs into your throat and recycled, broken down and recycled. And if you're sick, you cough to get rid of the stuff. And of course, that has the property of spreading the virus to other people, but it's also getting the virus out of your system. And there also are cells, macrophages and neutrophils that go around inside of your lungs, phagocytosing, eating the virus, generally destroying it. Sometimes they get infected themselves. HIV is an example of virus which infects the immune cells that are supposed to be getting rid of it. And in fact, one of the things that seems to happen in SARS-CoV-2 is that the macrophages and neutrophils that scavenge the virus then die. At least some of them do. So the scavenging doesn't work very well. Phagocytosis. So there's clearance chemically because the virus breaks down, because there are molecules in the first, the virus itself doesn't, even in perfect environment, doesn't last forever. So it breaks down on its own. Secondly, there's specific molecules in your mucus that are there to kill viruses, RNases and other things, antibodies. You don't have specific antibodies for the virus until about day 8 or 9 or 10. But there are non-specific antibodies that glom viruses together generically and get them, and those aggregated viruses can't infect. The viruses can be phagocytosed, and the active clearance in your lungs by the cilia is getting the virus out. As I say, the unfortunate thing is that when you cough, you're giving it to somebody else, but as far as your cells are concerned inside your lungs, it's gone. And so there are a whole variety of things that are going on simultaneously. They're clearing that virus. And so in our first models here, there was nothing that took virus out of circulation except in this model that I just wrote where the virus recombined with the cells. But in reality, the virus is cleared in your lungs. And the lifetime of the virus in the tissue is not trivial to estimate, but it's typically around 2 to 3 hours in your lungs, for example. And so let's say 2.5 hours. 2.5 hours, we're always having to take the reciprocals of things and we put it in days. So 2.5 hours means that you clear over 1/10 of a day, give or take. And so that means that your clearance rate is 1/10, so 10 per day. Got that? So if we're going to do decay, viral decay is about a rate of 9.4. And here I've cheated a little bit because I actually know what number I need. But if you use 10, it's not going to make a big difference. So why don't you try putting that in? So now I want you to add a decay term for the virus and use a decay rate to begin with of 9.4 per day. Means the virus sticks around for 2.5 hours. And I'd undo my equation here to do that. So is that, and I apologize that this equation is cut off here, but I think you can figure it out from that. Everybody got that? So why don't we all try that one together and add viral decay and see what it does. So we're going back basically to our earlier model. Well, do this one. Our earlier model here. I changed things around. So that was back to our original model without, I made a K. K was what? Before? K was, I've been changing things without keeping track. So somebody has to remind me. A K was the conversion time, and that was, we said 6 hours, so it was 4 per day. You should have it in your code. And then we were 2.5. That's back where we were. And so now add the decay of the virus. What should the decay of the virus do before you do it? What do you expect it to do? If I have viral decay, is it going to speed up my infection or is it going to slow down the infection?
Slow it down.
Should slow it down. And there should be a big difference, which is that instead of having the amount of virus go up without limit, at some point the virus amounts will saturate. And so here, the virus goes up very fast and then it slows down. But with the decay turned on, it should actually stay flat or even go down. So why don't you try that and see what happens? Okay, with that lifetime, the effect wasn't hugely dramatic, but now I really do get a flat curve and it saturates here at 10 to the 6th. And so if you remember my experiment, it really had a maximum of 10 to the 6th. And now I really have a maximum of 10 to the 6th, which is why I picked the number. The shapes of the curves and the timescales didn't change dramatically. If I make that decay constant much bigger, let's make it 10 times bigger. Slows everything down a lot, and I'll find that my maximum is also much less. My maximum is 10 to the 5th, factor of 10. If I make it smaller, so I make my viral lifetime much longer, the time scales for things don't change very much, but my maximum now comes up about 10 to the 7th. And notice here, I have that same rollover that I had before because it takes time for that to kick in. Then it depends also on the I1 transition. Okay. Did everybody get something comparable to that, or did you get something different? The other thing that we could do here that could potentially be interesting is put our plus V back in. I don't have that in my notes. Oops. What happened there? I pasted something rather than typing plus V. Well, here, because I don't have enough initial virus, nothing happens at the beginning. Slows things down a lot. What happens here? I still get my rapid decrease. I still get my exponential increase. I still get my speed up. When I lose all of my susceptible cells, I'm not taking virus out of my circulation anymore that way. And then I get this rock solid flat saturation at 10 to the 6th. notice that if I only measured my viral concentration once per day, I could not distinguish this shape from the other shape. So let me see if I can quickly put those two on top of each other. Sorry, I did this too fast. What I want to do is show you how they're different. So give me 1 second to do that. T loading all string 2, simulate, and we'll do a plot and we'll show false on the first one. And then we'll do the same thing for the second one. We'll have to reload it. We'll copy these. We'll do a show false. And we'll do model string 2 and see if that works. Oops. Invalid syntax. That's not very informative, is it? What did I do wrong? Show false doesn't seem to work, does it? Oh, I should do. Now, why is it? All right. I'll just plot it. I'm not going to do this. We'll plot them next to each other. Okay. So if I compare the two, the one with the viral combination is with the virus being used up by the combination is much slower. I have to rescale everything to see what's going on. So you have to notice the timescales are quite different. And if I look at my viral load, in the one with the viral recombination, it decreases very rapidly, increases in this biphasic way, and then saturates. In the one where I don't have the viral recombination, it still goes down early, but much more slowly because it's due to that decay term, not due to the recombination. It comes up. I don't get this acceleration when I run out of susceptible cells and I saturate. Notice that the saturation value is the same. And what's sort of interesting about this is if I did an experiment where I started out with a given amount of virus, and then a day later I made a measurement, And then one day after that, I made a measurement. I probably couldn't tell these two curves apart. Even though looking at them, they're very different. If I only make my measurement every 24 hours or 48 hours, I don't have enough information to tell them apart. And so one of the big reasons that people don't use the recombination T plus V term that I've just shown you is that we don't have accurate enough measurements of how the virus changes in time to be able to tell the two things apart. In that case, you might as well use the simpler one. All right. Okay. So the big result from what we did here is that we actually got the saturation to happen at the right value. Experimentally, if we started with a TCID, log TCID of 0 or 1, a value of 1, non-log, it saturates at 1,000,000. And for the parameters we picked, it saturates at 1,000,000. So now we have three things right. We have that initial delay of 6 hours. We have that exponential growth of 10 to the 6th over a day. And the thing stops after a day at a value of 1,000,000. And so these three equations together give us the first three phases of our viral load curve. And I want to emphasize that that's pretty surprising. The model that's simple, and we'll point out, as we've just done a fair amount of exploration of, that we know that we should be taking the virus out of solution, and it makes a big difference, that simple model actually works. If I wanted to get this more complicated model to work, where I take virus out of solution, then I'd have to retune my parameters. I could do it. What I'd find is that I get this squiggly curve here. How could I tell, looking at these 2 points, I have 3 points here, one at time 6, one at time 24, and one at time 2 days. Well, I'd have to rescale this viral load. I'd have to speed things up a little bit. I can do that if I need to. But with these 3 data points, could I tell the difference between this S-shaped curve here and this straight line? No, I couldn't. I wouldn't have enough information. And so this experimental measurement can't tell me which of these two models is better. I think it's worth thinking about that when you're doing modeling. It's nice to write a model that's realistic, but it's also sort of frustrating if the experimental data aren't sufficiently detailed to be able to tell things apart. If we wanted to make this viral load curve match better, what would we have to do? Well, we'd have to shorten this time scale for growth. The easiest way to shorten that time scale would be to make the conversion time kappa faster. I think if I made this something like 10, it might work. That speeded things up, not by much. It's still taking 6 days to reach its maximum. Here it's doing it after half a day. So I'd have to change other things as well to speed it up. I'd have to think more carefully to make that work. On the other hand, if I believe that I need to use T plus V here, because that's what's really happening, then the fact that the other model gives you the right curve is what's called a misfit. I've got a model that's wrong, but happens to be able to reproduce my data. And I think it's quite interesting that 95% of the papers, Josh, of all the papers you looked at in this field, how many have used T plus V goes to I1 versus just T goes to I1? None. So of all the papers he's looked at, none of them use the right relationship.
Well, what they would say is that that is implicitly represented in the decay of the virus.
Maybe. But the difference is the decay of the virus is independent of the number of susceptible cells.
Right.
And so you get a shape that's totally different, right? You get this versus that.
Mhm.
Curves are very different. And the infection pattern is different, quite different too. So again, I'm not trying to point fingers. I'm just saying that it's something to think about when you're building models. And I appreciate at some level, I have to say I appreciate your patience, because these are relatively sophisticated and advanced concepts that might not be covered in a course initially. But you seem like a very sophisticated bunch. And as engineer, if you're math bio, we might not consider this. As engineers, what we care about is that we get things right and that they work. And we want the model to represent the reality. And so I think it's important to remember that having a model that simply gives us the right curve, if the fundamental mechanisms embodied in the model are wrong, it's not very satisfying. And so as engineers, I think we ought to be thinking about why this is happening. Because even if we get the curve right here, if we change the parameter and the fundamental mechanism of the model is wrong, the dependence on the parameter will be wrong. And that's how you make predictions in engineering, where you build a bridge that falls down because you extrapolated wrong. And so it's worth thinking about that, I think. Okay. And so now with this viral load curve, we could play around a little bit with the saturation, and perhaps instead of going on to the next step, because we're almost out of time, why don't we just do a little bit of that together? So I'm going to put back what I had here. And that was model string 2. That's the one that I had the original model in. And why don't you just spend the last couple of minutes of the class exploring what happens when you change the infectivity, the eclipse time, and the amount of viral release and the viral decay term. In particular, you'll find that if you make the viral decay term 10 times bigger, the maximum amount of virus goes down by 10. You make the viral decay term 10 times smaller, the maximum amount of virus goes up by 10. But it doesn't just change the level, it also changes the time scales. So this is back to our original model. And now I'd like everybody to try just playing a little bit with some of these parameters. Again, in particular, you'll find, I think, that this changing the clearance time, here I'm making the viral lifetime 10 times longer. The final amount of virus goes up by 10. Timescales don't change much. Make the amount of the viral lifetime 10 times shorter. Basically slows everything down a lot. And if I made it run for longer so I could see what the equilibria are, the final amount of virus is 10 times less. It takes 5 days instead of 2 days to go through. Okay, are there any questions about all of this? Did it make any sense? Did it make sense at all? If it didn't, please complain. And I hope people found that at least a little bit stimulating. I think the homework we assigned is all right based on this. For once, we got through about what we expected. What I'd like to do next is something which we did sort of underhanded way in class, which is at the moment we have cells producing, susceptible cells becoming early infected, becoming late infected, producing virus. But we know if this were all that happened in your body, you probably wouldn't care whether you had a virus or not. You'd be coughing out virus and infecting other people, but you wouldn't really care because the virus isn't doing anything really harmful. And so the thing that we've, I don't know why tellurium keeps jumping around. I'm not touching anything in this window and it jumps around. I apologize for that. But we haven't included the fact that cells die when they produce virus. And so the next thing we're going to do in our modeling, and we'll do that next time, will be including cell death. Because in fact, the cell's typical cell, which is infected with SARS-CoV-2, will churn out somewhere between 3,000 to 6,000 viral particles, most of which are probably not infectious. The actual number of infectious viral particles from that TCID is more like 20 or 30. So that means that most of the virus particles the cell produces don't ever go on to infect anybody else. We don't know exactly why they're defective, but they are. Viruses are quite inefficient in this case. It's interesting because a single virus particle can give you a disease. But within the cell, when the cell is turned into a virus factory, like with flu, most of the virus particles that are produced are duds. So the quality control for viruses is very poor. Doesn't matter from the point of view of the virus as long as it can affect something. But we'll include the fact that the cells die. And exactly how long they live before they are killed by the virus and other things is not completely clear. But it's typically probably a couple of days. 2 days. For flu, it's not that long. It's probably 12 hours, 18 hours. And that's going to be the beginning of your problem when you get sick. Because you're going to start having cells in your lung that are dying. And those cells are doing things like maintaining the integrity of the lung, keeping fluid out of your lungs, transferring oxygen from the air into your blood. And so when you have a lot of cells in your lung dying, first you get leakage of fluid into the lungs, and then you get, which is pneumonia, and then you get loss of oxygen transfer from the air into your blood. So your blood oxygen levels go down. And that's why respiratory diseases, the beginning of why, one of the main reasons why respiratory diseases can kill you, although SARS also kills you in interesting ways like causing blood clots. But a model of a virus that doesn't have the cells die is pretty defective. And so, again, it's interesting that we're able to reproduce the beginning of this viral load curve without that. We're not going to be able to reproduce the later phases. And experimentally, in those cell culture experiments, cell death starts what? About in those plaque assays for influenza, when did cell death start?
12 to 16 hours.
12 to 16 hours. So it's a little faster. Flu is pretty fast. But for SARS-CoV-2, it seems to be about 2 days to 3 days before you get some cell death, maybe even 4. And it's also true that the way that the virus kills cells is a little more complex than we're going to get let on. Conceptually, we're going to say essentially that the virus is directly killing the cells. And that does happen. The virus doesn't have mechanisms to not use up as the cell produces exponentially more and more virus. More and more of the machinery of the cell is being used to produce virus and not the things the cell needs. and eventually that causes the cell to die. There also are active mechanisms where cells which detect virus inside of themselves commit suicide by apoptosis and pyroptosis. Mitochondrial permeabilization is one of the ways they do that. And then there are a variety of signaling mechanisms through interferon, which Josh has worked on, that also can lead to cell death. So they're actually, the virus can kill cells directly and indirectly in a whole variety of ways, some of which are protective. The cell is actually actively killing itself because it knows it's producing virus and it doesn't want to do that. In other cases, it's an accident. The virus simply is overloading the cell. But we're going to then, next time, start by including a cell death model and seeing what happens. And it's going to make a big difference. Everything will be quite different when we start including cell death. Okay, any other questions before we break? Josh, were you able to upload the homework assignment?
It's finished. I'll upload it now that we've finished the class. I was waiting to see.
Great. Okay, good. Thank you. And I will, was everybody able to get the demos to run all right? Did anybody have trouble running them today? Okay, and I will try to fix, there were a couple, I saw some typos in the slides, so I'll try to fix those. Please, if you have questions about projects, please do work on projects. Feel free to talk to me anytime about projects and progress. If I don't see you before next week, I will see you next week, I hope. And stay healthy. Try to avoid putting the simulations that we're doing into experimental action. All right. Okay. Thanks, everybody, and I'll see you next week.